am considering is, given the difficulty in giving exams, I'm ready to go all out. And what this means is a lot of work on my part, a lot of Zoom time on my part, in which, where I think what I'm going to do with exams is I'm going to give them one-on-one, where the person is interacting with me directly, asking questions and whatever, and we can I can then grade the person as they're going along, and if the person is having real difficulty, we could stop that exam at that point and reinitiate it at some other point. I don't know how many of you know other people who have taken the course with me. Some of the people who are here today are literally sitting through the course a second time. What I basically do is I have a full understanding that the learning curve for different people over a long period of time is different, so that I'm not the kind of person that gives an exam and then says, well, you got a 24, tough luck, that's it. Obviously, as it says in the syllabus, any grade under 80, you can retake the exact same exam, and the second taking an exam would count 2/3 versus 1/3, and I may even modify this further. What my total interest is in this course is not that you have everything memorized, but rather you show a basic competence in very quickly identifying things and understanding things. And this is how I'm going to lay out the course. So again, there is, what am I going to try to say? This under these very difficult circumstances, we have one more person coming in. I don't want to, there we go. Okay. Under these rather difficult circumstances, I totally understand this, and I will be working with you towards getting the exam. So what I'm going to do at this point is not talk about that now, but rather we will bring it up at the beginning of next class or the class after that, and I will give you further information as to how I'm going to be doing this type of examining. And that would probably start a week after I finish the spinal cord. Okay, as you can tell, we're moving at a rate, and sometimes I move a bit slower, and I want to make sure that you got this initial stuff, and then when I see the exam and I see how people are doing, I know how to titrate the time that I spend on things. So all of this is obviously recorded, so if there's anybody who had to go off or whatever, we can adjust and move on. So with that, I'm going to go down here, and what I have here is lecture 1, which we were on, and what we have here is and here we are with the structure. So here's the superior surface of the brain, and what I'm basically going to do is review this. So if people have questions or something as I review this, because again, there's almost 30 of you here, it's very hard to see this. I'm going to go through it. If you have questions for me to go on before I move off of this slide and go on to the next one. I want you to, hit the chat, put a chat message in, whatever. So, what we're dealing with here is the superior surface of the brain, and what we have is the interhemispheric fissure or longitudinal sulcus, and what we can see is the frontal pole, which is rostral, the occipital pole, which is caudal. The way you can differentiate the frontal and occipital pole is that in most human brains, the occipital pole is a bit wider, the sulcus, than the frontal pole. And then, of course, you can just see this by, if I reverse this, you would basically see a very clear superior frontal gyrus and middle frontal gyrus versus the way the occipital lobe is structured. So, the interhemispheric fissure or central sulcus, and then, of course, the other major sulcus that we're looking at here is the central sulcus or Rolandic fissure. And the central sulcus or Rolandic fissure, one of the major things that's contained within it is the middle cerebral artery. We're going to be going over the blood supply, and you'll see why that's important. And then, of course, the two adjacent gyri are the precentral gyrus and the postcentral gyrus. By definition, the precentral gyrus is in the frontal lobe, because anything rostral to the central sulcus on the superior surface of the brain is considered frontal lobe. And the precentral gyrus, of course, is known also as primary motor cortex. And conversely, the postcentral gyrus, which is in the parietal lobe, the most rostral structure in the parietal lobe, is immediately caudal to the central sulcus, and it is also known as primary somatosensory cortex. Okay. Now again, looking at the superior surface, the next important sulcus that we want to look at is the precentral sulcus. The precentral sulcus and the central sulcus obviously border the precentral gyrus or primary motor cortex. So whereas the precentral gyrus is organized moving from the superior surface and moving out laterally, the other major gyri in the frontal lobe on the superior surface are sort of layered horizontally. So what we have here is the superior frontal gyrus, then the superior frontal sulcus, and then the middle frontal gyrus. Okay? So the frontal lobe on the superior surface is made up, what you see is three major gyri here. Precentral gyrus, or primary motor cortex, the superior frontal gyrus, and the middle frontal gyrus. On the parietal lobe, what you're again going to see is the postcentral gyrus, and then caudal to that is the superior parietal lobule, and then the inferior parietal lobule. What you can also see on the superior surface, we see it also on the lateral surface, is the angular gyrus. So, if we are thinking of the parietal lobe on the superior surface, we have four major gyri: the postcentral gyrus, or primary somatosensory cortex, the superior parietal lobule, the inferior parietal lobule, and the angular gyrus. Then in the caudal portion of the superior surface, what we have is the occipital lobe, but the occipital lobe does not have a clear demarcation here. Okay, let me just stop sharing for a second and look at chat and see. Okay, someone just asked, and I see you're a new student, very good question. What's the difference between a sulcus and a gyrus? A gyrus is the actual tissue that we're basically looking at, okay? The actual brain structure, whereas a sulcus is the space or ridge or fissure between two gyri, okay? So, and someone said, think of the grooves, gyrus is bumps, sulci go in. You can think of it many, many ways, and of course what you can do is check out in the book, and of course Google all of this stuff. But, okay, let me see if there was any other questions here about that. Okay. On a vertical axis is supposed... Okay, no, on... yes, I think you're asking on a vertical axis as opposed to brains horizontally. Rostral is superior and caudal, rostral on the vertical axis, rostral is towards the head, caudal is towards the tail. Don't mix up rostral and superior and caudal and inferior. I see that note. Let's see if there's any other note. Okay, there we go. I don't see any other notes there. So, okay, we'll go back to share screen. And, okay, so that's the superior surface. And now, and again, I'm going to do this very quickly. Here's an actual brain section. Interhemispheric fissure, frontal pole, occipital pole, central sulcus or Rolandic fissure, precentral gyrus, precentral sulcus, superior frontal gyrus, superior frontal sulcus, middle frontal gyrus. Then we go to the central, we start the central, and all of that's frontal lobe. Doing the central sulcus, anything caudal to this is parietal. So we have the postcentral gyrus or primary somatosensory cortex. We have the superior parietal lobule. We have the inferior parietal lobule. And then we have the angular gyrus. Okay? And then we finally have the occipital lobe with no clear demarcation between parietal and occipital on the superior surface. So now we're going to the lateral surface that we covered the last time. And here we're looking at the lateral surface, and obviously if I were on an old TV show called Alfred Hitchcock Presents, he would come on to the, there would be a silhouette of Alfred Hitchcock, and his face in shadow would come and fill up the thing. So, of course, I'd be facing this way, okay? Why? Because this, again, on the horizontal axis is rostral, this is caudal. So, obviously, we don't see the interhemispheric fissure because we're on the far lateral surface. But what we do see is the central sulcus, which, of course, started out by the interhemispheric fissure, went laterally, and now you can see that central sulcus is moving all the way down right here to another sulcus, which of course is the lateral sulcus, or what is known as the Sylvian fissure, Sylvian. And again, if we think about a sulcus as having some functionality, one major aspect of functionality that you'd be basically looking at is blood supply. And here is how the middle cerebral artery enters into the lateral sulcus, and then it goes up the central sulcus to vascularize an enormous part of the brain that you basically can see here. Now, and so now, if we just review our frontal and parietal lobes first, what we can see is the central sulcus. So anything now on the lateral surface that is rostral to the central sulcus, yet superior to the temporal sulcus, is defined as frontal lobe. So what we have are the following gyri. Now we're worried about 4 major gyri in the frontal lobe on the lateral surface. We have the precentral gyrus, otherwise known as primary motor cortex. We then have the precentral sulcus, and then we have 3 gyri that are stacked horizontally on top of them, rostral to the precentral sulcus. We have the superior frontal gyrus that, of course, we saw on the superior surface. We have the superior frontal sulcus. We have the middle frontal gyrus. We have the inferior frontal sulcus. And then we have this massive area here that we basically call the inferior frontal gyrus. And the inferior frontal gyrus, because of its size, is subdivided into 3 parts. So when you are going to identify this, you're not going to basically just call it pars something. You're going to call it inferior frontal gyrus, comma, pars opercularis. Sorry about that. Opercularis right here. And the hint there is an operculum is Latin for slit. So you can see that sort of slit-like feature there, the pars opercularis. Then the pars triangularis. Why it's called triangularis, I don't know. You'd have to ask the Latin neuroanatomist who named it. And then the largest part is, of course, the pars orbitalis. And the inferior frontal gyrus pars orbitalis is basically known for that simply because what you have is the eyeball right here, and it's the area just above the eyeball. Now, I just want to point out something here. And again, why it's important to study sulci and why it's important to study functionality. If you remember, you have the middle cerebral artery, which is running through the lateral sulcus, and then it runs through the central sulcus. Now, think of one of the major forms of a stroke, okay? When a person has a stroke, very often it is because there is an occlusion of either the left or the right middle cerebral artery. And if you have a stroke in the middle cerebral artery, let's say on the left side, it's going to manifest itself on the right side of the body, the contralateral side. And the reason for that is because the pathways, the sensory and motor pathways that control the right side of the body, a large number of them either completely or almost 95% decussate, okay? And I'm going to explain that later. They cross the midline. So therefore, what you are getting is that. So all of a sudden, look at what happens here. The pre and post central gyri can be affected by a stoppage of blood of the middle cerebral artery. What else can be stopped? Well, what's another major thing that can happen? For people who are right-handed, very, very right-handed, 95% of those people have their receptive and expressive speech on the left side of the, controlled by the left side of the brain. So if on this left side we had an infarct of the middle cerebral artery, what would happen is blood to the inferior frontal gyrus pars orbitalis would be interrupted, and blood to the superior temporal gyrus would be interrupted. And what did I point out in the last class? That this inferior frontal gyrus pars orbitalis is also known as Broca's area because it appears to mediate what is called expressive speech, and loss of function of the inferior frontal gyrus pars orbitalis will result in something called expressive aphasia, the inability of the person to express speech. And very often, the only kind of speech they're able to express, if they're very, very severe, is where they get very, very emotional and they're trying to get out. And what kind of words usually come out? Usually curse words, okay? It's very interesting. Okay, and then on the other hand, what we identified in the last class as the superior temporal gyrus is also known as Heschel's gyrus, and is also known as primary auditory cortex. And this is an area identified by the neurosurgeon Wernicke, who basically found a patient who had a lesion restricted just to this area, and they suffered from receptive aphasia, the inability to understand speech. Now, most of the time, if there is a major damage to the middle cerebral artery, it will affect the inferior frontal gyrus pars orbitalis, expressive aphasia. It will affect the superior temporal gyrus, receptive aphasia, and then the interruption would persist up through the central sulcus and the precentral gyrus. Motor problems on the contralateral side of the body would be affected, and the postcentral gyrus, the somatosensory cortex. Where's my pointer here? Come on. Where the hell my pointer go? There it is. His primary somatosensory cortex, and the people would suffer sensory deficits. So what you can basically see is very importance of this blood supply. And of course, what is the middle cerebral artery? The middle cerebral artery is the logical continuation of the internal carotid. The internal carotid is the major blood supply. We're going to be reviewing this coming from the carotid artery. And the carotid artery is the first major artery that comes off the aorta. So if you go all the way back to my first slide back in of last week, where we basically saw that 17 to 20% of the oxygen and blood flow runs to the brain, you can see how important these things are, and you can see how an interruption of that can have dramatic effects. So now let's continue with the parietal lobe, and what we see here is the central sulcus, the postcentral gyrus, the superior parietal lobule, the inferior parietal lobule, and the angular gyrus. So those are the major structures in the parietal lobe on the lateral surface. And of course, the parietal and occipital lobe do not have a clear sulcus demarcating them. So now, The point is, we define the frontal lobe now as on the lateral surface as being what? Rostral to the central sulcus, superior to the lateral sulcus. The parietal lobe is defined as superior to the lateral sulcus and caudal to the central sulcus. So now this brings us to the temporal lobe. The temporal lobe is any portion that is inferior to the lateral sulcus. And now, whereas before with the frontal lobe, I pointed out that there were 3 major gyri, which means there are 2 sulci. With the temporal lobe, there are 4 gyri, which means there are 3 sulci. So we're going to be naming the 3 sulci the superior temporal sulcus, the middle temporal sulcus, and the inferior temporal sulcus. The point is, however, on the lateral surface of the brain, we only see 3 of the gyri. We see the very, very large superior temporal gyrus, otherwise known as primary auditory cortex, otherwise known as Heschel's gyrus, and otherwise known as Wernicke's area, all different levels of functionality. We then have the superior temporal sulcus. We then have the middle temporal gyrus. We then have the middle temporal sulcus. And we can see a little piece of the inferior temporal gyrus. What happens to the inferior temporal gyrus? It continues on the inferior surface of the brain, and then it will continue up onto the medial surface of the brain. So in a couple of minutes, we're going to be looking at the inferior surface of the brain, and we're going to be seeing that inferior temporal gyrus there again. Okay? So that's the temporal lobe. Again, the occipital lobe, we're not making big definitions here, simply because we don't have clear sulci. I'll talk about them later. Okay, now below this on the lateral surface, what do we end up also seeing? What we also end up seeing is subcortical structures, and of course we're going to be seeing things like the midbrain, the pons, and the medulla, which I'm going to get into in a minute. And then we see this big cauliflower-like structure on the lateral surface, which of course is the cerebellum. Okay, so now we're going to shoot past this and go to our, here we go, looking at the actual structure. So again, the 2 major sulci in the lateral surface is the, central sulcus, and the lateral sulcus. Then we have the precentral gyrus, the superior frontal gyrus, the middle frontal gyrus, and then the extremely large inferior frontal gyrus, which is made up of the pars opercularis, right here, the pars triangularis, and the whole rest of it is the pars orbitalis. Then on the parietal lobe, we have the central sulcus, we have the postcentral gyrus, we have the superior parietal lobule, we have the inferior parietal lobule, we have the angular gyrus, then we have the occipital lobe, and then on the temporal lobe, we have the superior temporal gyrus, the middle temporal gyrus, and the inferior temporal gyrus. And then we see some subcortical structures and cerebellum. So now, we are now going to look at the inferior surface of the brain. And what I did last week was cover two major general characteristics about the brain that you can basically infer from the inferior surface. The first thing is encephalic development. And what we're doing here is by the dots, what we are pointing out is the development of the brain at the level of around the late second trimester prenatally. So what we basically see here is a prosencephalon, which is the most rostral portion, the mesencephalon, which is the middle area, and then the rhombencephalon, which is the most caudal area. So basically, if we're thinking somewhere in the late second trimester, what we have in that fetus is what? You have a spinal cord, and then you have the three most rostral portions of the nervous system has now differentiated into a rhombencephalon, into a mesencephalon, and into a prosencephalon. Then during the third trimester of fetal development, what we basically now do is expand from three different levels of the brain up to 5 different levels. The rhombencephalon differentiates further into the myelencephalon and the metencephalon. The mesencephalon pretty much stays the same. And then the prosencephalon differentiates into a more caudal diencephalon and a more rostral telencephalon. So now, all of a sudden, we have birth and we have postnatal development. And what is now happening? The myelencephalon is eventually going to develop into the medulla and the cerebellum. The metencephalon is going to develop into what we call the pons. The mesencephalon is going to develop into what we call the midbrain. The diencephalon is going to develop into what we end up calling the thalamus and hypothalamus. Look at the word. It's very clear that the hypothalamus, hypo meaning below, is inferior to the thalamus. Just a little hint, okay? And then the telencephalon really develops into the cortex, all 4 lobes, a structure called the basal ganglia, and another series of structures called the limbic system. So, when we look at encephalic development, as we did in the near the end of the last class, what we basically see on this inferior surface of the brain is a rhombencephalon. Here is the outline of the rhombencephalon. All of this is derived from rhombencephalon. The mesencephalon is this area right here. The mesencephalon, the rostral border of the mesencephalon, is these 2 egg-shaped structures called the mammillary bodies. M-A-M-M-I-L-A-R-Y. Okay, we'll be talking a lot about them as we move along. But that is the mesencephalon. Then the telencephalon goes everywhere from the mammillary bodies forward and covers all of this cortex here. Then, of course, in the third trimester, what we pointed out is that the rhombencephalon differentiates. It now becomes the myelencephalon and the metencephalon. The mesencephalon stays the same. And now the diencephalon, the only part of the diencephalon that we can see here, where's my damn pointer go? There it is the hypothalamus area right here. And then the telencephalon is everything else. And of course, in postnatal development, we now see in the telencephalon the cortex. That's about the only thing we see on this inferior surface of the brain. We see the hypothalamus and the pituitary area for the diencephalon. We see what is called the cerebral peduncle. I'll be going into this in detail in a minute. The inferior surface of the midbrain. We then see a middle cerebellar peduncle, which covers the inferior surface of the pons, and then we see the medulla and we see the cerebellum. Okay. So now the other things that we covered here and sort of finished with was looking at the 12 cranial nerves, and what I pointed out is that for each of the 12 cranial nerves, what you should know is 6 pieces of information each. Obviously, the cranial nerve number, which is given, and by the way cranial nerve numbers are given is a rostrocaudal direction. So the lowest number is the most rostral. Then, of course, you associate the number with the name of the cranial nerve. You then should know at what level of the encephalic development in the brain is the cranial nerve. And then we realize that there are 3 types of cranial nerves. There is a purely sensory type of cranial nerve, and that would be also using the synonym afferent. So a sensory afferent is a cranial nerve in which you're coming from outside the brain and going into the brain. And then the 6th piece of information you want to know is whether or not the cranial nerve is entering or exiting medially or laterally. Now that might sound like a trivial piece of information, but it's going to become invaluable when you're looking at different brain slices as we move through the brain, and we can identify the cranial nerves and then identify exactly where in the brain we are. So the first cranial nerve is the olfactory. Oh, and the 6th piece of information is what is a one word or a couple of word function for that cranial nerve. We're going to go into a lot more detail as we encounter each of these cranial nerves. So we have the olfactory, which is in the telencephalon, sensory, smell, and it's entering medially. We have the optic nerve in the rostral diencephalon. It's sensory, it's involved in vision, and entering medially. We then have the 3rd cranial nerve, the oculomotor, which is found in the middle of the mesencephalon. It's a purely motor. It controls a bunch of different eye movements, different eye muscles, and it exits the brain medially. The 4th cranial nerve is the trochlea. It's in the caudal mesencephalon. Again, it's the 2nd of the 3 cranial nerves that control eye movements, but it weirdly exits the brain laterally and dorsally. We'll see this in a little while. The 5th cranial nerve is the metencephalon. It is a mixed cranial nerve, which means what? It has a sensory component and it has a motor component. And one major thing that it's involved in is mastication, mouth movements, facial movements, etc., it has a lateral path. It's the second largest cranial nerve. The sixth cranial nerve is the abducens. It's found on the mesencephalic myelencephalic border. It is the third of the three cranial nerves that control eye movements, and it exits the brain medially. So here we can see these six cranial nerves. What we can see right here is the olfactory. Then we can see the optic. And as I pointed out in the last class, the optic nerve, whereas the olfactory, all of the cranial nerve goes into the side in which each nerve got the information. With the optic, what is basically happening here in a human being, there's a 50% decussation of optic fibers, so that information from, obviously, information going into one eye contains the whole visual field, and then information from both eyes go back to the occipital cortex and manifest themselves. We'll talk about that later. The third cranial nerve is the oculomotor, the first of the eye movement. The fourth we can't see because it's exiting the brain dorsally and laterally. That is our friend the trochlea. The fifth cranial nerve, the second largest of the cranial nerves, is right here. That's the trigeminal. It is mixed, having both sensory and motor components. And as I pointed out, the trigeminal has three different branches that innervates the ophthalmic, maxillary, and mandibular portions of the face. And then the sixth cranial nerve is the abducens, and that is the third of the three cranial nerves that control eye movements. Then cranial nerves 7 through 12, all of them are found in the myelencephalon in the medulla. Okay? And what we have here is the seventh, facial, which is a mixed cranial nerve and is involved in taste and a whole bunch of facial movements. It has a lateral path. The vestibular cochlea is sensory, and it's involved in audition and balance and coordination, and it enters the brain laterally. The ninth cranial nerve is glossopharyngeal. It is a mixed cranial nerve, both sensory and motor, and modulates the larynx and the pharynx, again having to do with digestion and also having to do with vocalization, lateral path. The 10th cranial nerve, the largest of the cranial nerves, is a mixed cranial nerve, both sensory and motor. And the way the vagus is so large, it basically controls the trunk. And if we use the diaphragm as a borderline, what we can see is something called the supradiaphragmatic vagus, which of course is the upper chest here, which of course is very much involved in respiration. And then the subdiaphragmatic vagus, which is below the diaphragm, and that is involved in digestion. And that also has a lateral path. Then the 11th cranial nerve, it has the distinctiveness of being the smallest, is a motor cranial nerve that controls an upper part of the chest here called the branchiomeric arch, and basically it has to do with inspiration and expiration of breath. And then finally, the 12th cranial nerve, the hypoglossal, is a motor cranial nerve. It's the most caudal, and it controls the posterior two-thirds of the tongue, which of course makes it very important in the process of swallowing. So if we now look at the 7th through 12th cranial nerves, what we basically see here is the 7th. Here is the 8th, showing both the vestibular and cochlear branches. The 9th cranial nerve is right here. The 10th, the largest cranial nerve, the vagus. The 11th, the smallest cranial nerve, the spinal accessory. And in fact, the spinal accessory is so small, it shares the same foramen or hole as it exits the skull as the vagus. And then the 12th cranial nerve is the hypoglossal, exiting the brain medially, as you can see in the medulla. Okay, so now let me just stop this for a second. And now basically look at particular questions. Looks like I'm getting a lot of comments here. Wow, okay. Seeing a lot of comments and a lot of people sort of helping each other. So superior, rostrum is beak in Latin, Spanish, okay. For inferior, think of a shark, okay. All right, you're looking up in the brain, all right. Okay, middle cerebral artery, circle of Willis, we're going to talk about that. Inferior, on the inferior is that dark structure part of the telencephalon or diencephalon. It's right above. We're going to be talking. That's part of the dark structure is going to be rostral to that is going to be the hypophysis or pituitary gland. We'll talk about that. Same. Thanks. The whole course. Do we have to know all those nerves? Do we have to know? All its nerves? No, you don't. You just have to basically, I would hope that you would remember it because it's a very important thing. Whenever people ask a question, I'm very often saying, is this going to be on the exam or not? Obviously, everything I'm saying, I hope you walk away with so you know for the rest of your life. But for the exam, just knowing those 12 by 6 would be very helpful. I'm just trying to give you more information. Okay.
No, that's fine. It was just a lot of information, so I missed a few.
Of course, it's a lot of information, and that's why we're doing a full review here, and I sort of want to make sure that these things are clear to you as best as they can be. And of course, if we want to, we can have asynchronous meetings in which I can really go over it if there's something that you don't know here. Okay, so now I'm going back to share screen, and we're back now, friend. Okay, so now, here is. I found this slide to be one of the most interesting types of things to basically show you a cranial nerve schematic. So if you take the 12 cranial nerves and divide them into 3 major types, you can see 1, 2, and 8 are sensory, 1 having to do with smell, 2 having to do with vision, 8 having to do with audition and balance. Then the motor nerves are 3, 4, and 6, and they are basically controlling all of the different eye muscles. We're going to be talking about 6 different major eye muscles, and then a 7th interesting eye muscle, actually even an 8th. So we're going to be talking about the 6 eye muscles that move the eyes. We're going to be talking about a huge eye muscle that makes the pupillary response, that colored eye muscle in the middle of your eye called an iris muscle. And then we're going to be talking about a muscle that controls your eyelid. And if you have a condition called ptosis, ptosis, that's called the droopy eyelid. Simply, you lost the motor control of that eyelid, and the eyelid goes to a relaxed mode, and it closes the eye. That's terrible. Okay, and then what we have, those are the three cranial nerves involved with the eyes. Then we have two more cranial nerves, which are purely motor, which is 11 and 12, controlling the posterior two-thirds of the tongue, the throat, and whatever. And then we have four cranial nerves, 5, 7, 9, and 10. And you can see where the thing is. By the way, does everybody see the interesting number 8? For Mr. I always found that to be cute. And you can see the 7th cranial nerve over here. You can also, that's the first cranial nerve, the 7th. And you basically can see where these things are controlled. And then I know, and here you basically can look from rostral on the top to caudal down at the bottom. And basically you can see how these afferents and efferents are being controlled in this slide. Okay, so now another quick area, we're going to go through this when we go into the diencephalic, when we talk about the hypothalamus. But what we're going to be talking about next here is the hypothalamo-hypophyseal portal system. That's a lot of syllables. So hypothalamo is what? The hypothalamus is the inferior portion of the diencephalon. Then we have the all-important hyphen. And what does a hyphen tell you? What comes before the hyphen is where it's coming from. What comes after the hyphen is where it's going to. So here what we are seeing is the hypothalamus is controlling the pituitary gland. It is at this point a unidirectional thing. And what we also then have is the interesting word portal, P-O-R-T-A-L, which basically talks about blood and blood flow, and specifically venous blood flow, blood running back away from the brain. So what we have literally is I always remember a favorite book that I read, my two sons who are now in their mid-30s, called Scruffy the Tugboat. And in Scruffy the Tugboat, some little kid is around where I am, upstate New York, and drops the tugboat into a stream. And all of a sudden, the little tugboat goes riding along, and now you're following the thing from the point of the tugboat. And where I live, by the way, my area, my water supply can run north because it's running downhill. I'm up high along Schoharie Creek, that then goes into the Mohawk River, that then goes into the Hudson River, that then goes into the Atlantic Ocean. Going south, I have the headwaters of the Delaware River that runs west of Jersey and out at the Delaware-New Jersey border. And then the third is a thing running west, which forms the Susquehanna River that runs out in Virginia. So in this story, the tugboat goes flying along, and it rides through Schoharie Creek, and it runs through the Mohawk River, it runs through the Hudson River, and just as it's passing the Statue of Liberty in Brooklyn, just before it's going into the Atlantic Ocean, the kid's grandfather pulls the little Scruffy the tugboat out and puts it back in the kid's bathtub. But the point of the matter is, forget about being tugboats. Think about substances, blood-borne substances. And the blood-borne substances that are produced by the hypothalamus, a number of hypothalamic structures, are what are called releasing factors and inhibitory factors. And these factors began to be discovered in the late 40s, and the last of the big guys, which was corticotropin releasing factor, was discovered in 1981. And it was discovered by two guys who originally collaborated with one another and then became rivals. And their names were Andrew Schally and Christian Guillemin. And Schally and Guillemin won the Nobel Prize for the understanding of how hypothalamic substances are then released into the bloodstream and affect what is called the adenohypophyseal system. Okay, the adenohypophyseal system is a blood-formed system, and basically here you have substances released into these by nuclei that are called parvocellular, and these parvocellular cells in the hypothalamus will send fibers down into a structure called the median eminence zona externa. And you can then release hypothalamic releasing or inhibitory factors like luteinizing hormone, releasing hormone, into the hypothalamo-hypophyseal portal system. And this portal system bathes the anterior and intermediate lobes of the pituitary, and they then act on pituitary cells to release a pituitary hormone like luteinizing hormone, and then that is then released into the blood that then acts on a target like the ovary. So here, how does a woman produce progesterone? And how does she increase the amount of progesterone? It's because of particular cells in the hypothalamus that secrete LHRH, release it into the blood, bathe the anterior lobe of the pituitary to make those particular cells that produce LH to release LH into the blood that then acts on a target, the ovary, to build up progesterone, and we'll talk about that later. But what you're basically seeing is an interaction among three different systems: a neural system, specifically in the hypothalamus, a blood-borne system, that is the hypothalamo-hypophyseal portal area, and then a hormonal system, which is the pituitary, that then releases again out into the circulation things that are going to be basically controlling a lot of our functions. Now, if there is an anterior and intermediate lobes of the pituitary, there obviously then is a posterior lobe of the pituitary. But the posterior lobe of the pituitary is not hormonal. It is a direct link and continuation of the brain. So therefore it is called the neurohypophyseal system. And what we're going to be seeing is that there's going to be magnocellular, oh my God, a very complex name. No, these are just large cells in the paraventricular and supraoptic nuclei in the hypothalamus that send their axons to the median eminence zona interna, and then the axons continue out through a very small little stalk that we're going to see called the infundibulum. And the infundibular stalk will then allow whatever is being produced by the paraventricular and supraoptic nuclei to continue down into the posterior lobe, where they sort of end up in the posterior lobe. And you almost think of the posterior lobe as this very large bouton in a synapse. And here are some substances that are released by these cells of the neurohypophysis: vasopressin and oxytocin. And there's a host of other substances. I am not going to get into the functionality of this, but on the inferior surface of the brain, we end up starting to see the brain here. So if we look at this, here's our friend the medulla. Here is the cerebellum. So medulla, cerebellum. Then here we have the pons. Then here we have the midbrain. Then remember those two egg-like structures? There's the mammillary bodies. Here is the optic nerve. Here is the optic chiasm. Here is the optic tract. And right between it, right in D right here, pointing at D, is that little thing called the infundibulum that's going to the posterior pituitary. And what you can basically see right around D is the underlying hypothalamus. Right here, they literally snipped off the end of the pituitary, so you wouldn't see that. But this is this tiny little area that we're dealing with, which basically is the major area for the whole field of something called neuroendocrinology. Okay, so now that was looking at a tiny area. So we've gone through encephalic development by looking at the inferior surface of the brain. We have gone through looking at the cranial nerves, being able to visualize 11 out of the 12 cranial nerves on the inferior surface of the brain. We then looked at the hypothalamo-hypophyseal axis on the inferior surface of the brain. Now what we're going to do is we're going to look at what are called cerebellar peduncles. Now what we're only going to be able to see is one of them on the inferior surface of the brain, but given that I'm telling you one, I'm going to tell you all three right now. So, going from rostral to caudal, we're going to see the superior cerebellar peduncle, then we have the middle cerebellar peduncle, and then we have the inferior cerebellar peduncle. Okay? The superior cerebellar peduncle is otherwise known as the brachium conjunctivum. And whenever you see the word brachium, it is basically fibers that are crisscrossing the brain. And they're crisscrossing the brain medially laterally. They're crisscrossing the brain medially laterally. So the superior cerebellar peduncle is going to crisscross, it's going to decussate, and it is called the brachium conjunctivum. And the superior cerebellar peduncle has reciprocal connections between the cerebellum and two very large rostral structures, the thalamus and the basal ganglia. So this is a major way that the cerebellum sends information and receives information from the thalamus and the basal ganglia. The middle cerebellar peduncle, which we're going to be looking at right now, is also known as the brachium pontus, which is no big surprise because when we were looking at the inferior surface of the brain, the only thing we see in the pons is our friend the middle cerebellar peduncle. And again, a brachium again is a crisscrossing, and it's an enormous number of fibers that are going from the one side of the cerebellum to the other side of the cerebellum, but in the meantime are interacting with hundreds of millions of neurons in the ventral pons called the Grecium pontus. We'll see that later. But the middle cerebellum or cerebellar peduncle, or brachium pontus, has these connections that interact the ventral midbrain, the pons and medulla. And then we have the inferior cerebellar peduncle, or restiform body. Now, this will come up in our future lectures on the spinal cord, because the inferior cerebellar peduncle, or restiform body, carries the major afferent connections from the spinal cord up to the cerebellum, with one exception. But the major amount of afferent connections from the spinal cord going into the cerebellum enter via the inferior cerebellar peduncle, or restiform body. Okay, so now notice we use the word called peduncle. And a peduncle, as I pointed out, is a big, large fiber tract. We saw them with the cerebellar peduncles. Now what we have is the cerebral peduncle. And we have the cerebral peduncle and that wonderfully named structure in the ventral medulla called the pyramids, okay, or the pyramidal tract. Now, and you'll start to discover why this is. And when we are talking about the cerebral peduncle and the pyramids, we are talking about the major efferent tract that is controlling the spinal cord. It is called the corticospinal tract. Okay? So, we saw the beginning of the corticospinal tract in our first look at the superior and then the lateral surfaces of the brain. Why? What did we see? We saw the precentral gyrus. And the precentral gyrus is found in the telencephalon. It is the caudal portion of the frontal lobe. And as I pointed out, it is somatotopically organized, so that if we looked on the superior surface of the brain and medially, and we started to stimulate that area, what we would start to see is movements in the lower part of the body. So the feet and the legs are on the superior surface, and then as we go out onto the lateral surface and go down, we then follow the legs, the trunk, the arms, the neck, and the head. So there is a somatotopic organization, and that the information on this one side of our head, right here, this left side, is basically exerting motor control on the right side of the body. The information on the right side of the head is exerting motor control on the left side of the body. Why? Because what we're going to notice is that there is going to be some degree of decussation. So, now what is interesting about these cells that make up the precentral gyrus? A vast majority of those cells are called BETZ, capital B-E-T-Z cells, or pyramidal cells. And that the vast majority of these cells look like upside down upside down triangles. So the tip of many of these cells is facing ventrally, okay? The top, which has the pyramid, very often has a one dendrite, and it's called an apical dendrite. A-P-I-C-A-L, apical. So it's almost like a tree going up, and it goes through the other cortical layers where these pyramidal cells, and it's getting input. So now this pyramidal cell finally gets to the point of firing. We'll talk about how that happens later on. And it will activate an axon. Virtually all of the axons of the corticospinal tract are type 1 motor efferents, which means they are heavily myelinated, which is the myelin sheath. And of course, when you have, the more myelinated the axon is, the faster it conducts information. So now we're going to take a journey from the precentral gyrus in the telencephalon and get all the way down into the whole spinal cord, and then that is a very long trip. So these axons travel long distances. They are very fast, and the first place they travel is to it's going to be called, we're going to see it, it's going to be called the internal capsule. And the way I describe the internal capsule when we are going to look at this in the brain later is, think of it like the Champs-Élysées in Paris, a grand boulevard of 10, 12 lanes of input that's going in and input that's going out. Now, think about this. There's going to be two portions to the internal capsule. One is the thing we're talking about now, being part of the corticospinal tract. And it is carrying motor output. So what part of where is, if you think of somatosensory input and motor output, what have you learned already? You learned that the precentral gyrus motor is rostral to the postcentral gyrus somatosensory. So now, if we just follow it, and we now are looking at the axons exiting the precentral gyrus, it's exiting the precentral gyrus through the internal capsule, which is, and it would be exiting through what we call the anterior limb of the internal capsule. Because what's going to be in the posterior limb of the internal capsule? The somatosensory input. Just like the Champs-Élysées go in two different directions, the anterior limb is taking corticospinal output, and the posterior limb is taking somatosensory input. So now, what is basically happening? The internal capsule is moving from the superior and lateral surfaces of the brain, and it's moving deeper into the brain, more inferiorly. So it is moving superior to inferior. So now I'm going to stand up, and I'm hoping that you're going to be able to see me here. So oh, damn it. I'm trying to let me put two pillows down so we're able to see this. Or actually, I'll just lean this down further like that. Here we go. Now you should be able to see it. Everybody see my leg? Here is the internal capsule. Thinking if I'm going this way, I'm going what? Superior to inferior. Now, where do I want to go? I want to go into the spinal cord. Otherwise, it hit the bottom of the brain and nothing would happen. So what do I have to do? The spinal cord is going this way. Okay? So what's basically happening? What I'm having here is a turn. Okay? So now let me describe what the turn is. Look at that word after internal capsule. Cru cerebri. Cru has to do with the knee. Okay? And it's basically the turning of the fibers moving. It moved superior to inferior through the diencephalon, and now it's turning so it can go on the inferior surface of the brain via the cru cerebri. So now, once we made that turn, we're now in the mesencephalon, and we call it the cerebral peduncle. It continues, these axons continue, and they get down, they now go through the pons, but something happened in the pons. This big belt of fibers, the middle cerebellar peduncle took up the inferior surface of the brain, so now this corticospinal tract is buried within the middle cerebellar peduncle, but keeps on going ventrally through the mesencephalon pons. And then what you're basically going to see in the medulla is the pyramids, which are the same corticospinal tract. Now you might go, well, why don't we just call it corticospinal tract, corticospinal tract, corticospinal tract? Well, back in the old days when there was lots of traffic in New York City, and every 8 minutes they would have a traffic report, and let's say they said, let's imagine Manhattan, and let's imagine where the Battery is. And if I travel north, I would go over the FDR at East River Drive. I would go up to 125th Street, and I would go up the Harlem River Drive. Then I'd get on the Cross Bronx Expressway going west a little, and then I'd get on the Henry Hudson Parkway and the West Side Highway going south, and eventually I'd get back to the Battery. What I could call that is the Circum-Manhattan Parkway. And if somebody actually went and said, hey, there's an accident on the Circum-Manhattan Parkway, everybody listening to that radio report would start cursing and going, where? That if you hear that there's the accident is on the FDR Drive and a specific portion of it, you know it, then da-da-da. The same thing happens here, that we can basically say if there was damage, is there damage in the precentral gyrus itself? Is there damage in the anterior limb of the internal capsule? Is there damage at the level of the cerebral peduncle? Is there damage at the level of the cru cerebri? Or is there damage in the pyramids? Now, at the pyramids in the rostral medulla, all of the fibers are still ipsilateral, right? From the right precentral gyrus in the rostral medulla is still there. But now, in the caudal medulla, we have this major crossing of fibers, the pyramidal decussation. Now, one of the decussation we talked about with the optic tract, and we saw its functional significance, is the optic tract has a 50% partial decussation. The pyramids have a 95% decussation. Okay? 95% of the fibers are going to decussate, and they will eventually go into something called the lateral corticospinal tract in the spinal cord. 5% of the fibers, remember, on the ventral surface of the brain, 5% of the fibers don't decussate, and they go directly into the spinal cord, and they become the anterior corticospinal tract. Now, this is a very major thing for recovery of function, because what basically was always argued was, oh, after a stroke, you sort of learned if the stroke was ipsilateral on one side of the brain, oh, we learned through some kind of recovery of function, things came back. No. Very often what it is those residual fibers that didn't decussate take over, not perfectly, but it takes over partially that motor function. So now, let us look at the cerebral peduncle and pyramids as part of the corticospinal tract in this inferior surface of the brain. So, what we can see right here is look at the size of the cerebral peduncle in the midbrain. And then, what's rostral to this? Well, remember, you're seeing hypothalamus here on the inferior surface of the brain, right? But you're not seeing internal capsule because it hasn't reached the inferior surface yet, and it's going to turn somewhere above this, superior to this, superior to this would be the crus cerebri and the internal capsule. And then, of course, what did I tell you? These cerebral peduncle corticospinal fibers keep going in this direction, but you can't see them in the pons because we have this big cummerbund like middle cerebellar peduncle covering them. They're still there, they're still going down, and they emerge on the inferior surface with the pyramids. And then right here is the pyramidal decussation, where 95% of the fibers cross, and they go in, the 95% of the fibers that cross go into the spinal cord as the lateral corticospinal tract, and 5% of them stay ipsilateral and form the anterior corticospinal tract. So that is the cerebral peduncle. We've seen the cerebellar peduncle. We've seen the encephalic development. We've seen the hypothalamo-hypophyseal portal system. We've seen the cranial nerves. So now let's start to look at, on the inferior surface, some of the other stuff. And what we have here in the frontal lobe is we have the orbital gyri, because we're on the inferior surface. The only thing we can see is the inferior frontal gyrus, and specifically the most inferior portion of the inferior frontal gyrus, which is the inferior frontal gyrus pars orbitalis. But something else we basically can see is the longitudinal fissure. Because remember, the longitudinal fissure we saw on the superior surface of the brain, but it goes through the frontal pole and extends on the inferior surface of the brain until we get to the diencephalon. Then in the temporal lobe, remember when we looked at the lateral surface, what were we able to see? We were able to see the superior and middle and a little part of the inferior frontal gyrus. On the inferior surface, we're going to see the inferior temporal gyrus and the parahippocampal gyrus. Then we're going to see a bump called the uncus. And then the final thing we're going to look at again is wonderful things called pyramids and olives. So, here is the frontal lobe. Here is the interhemispheric fissure coming through the frontal pole, and then continuing on the inferior surface of the brain until we literally get up to where the optic chiasm is. Now again, if you remember when I said the lateral surface of the brain was carrying an important blood vessel called the middle cerebral artery. The interhemispheric fissure on the inferior surface of the brain carries another very important artery called the anterior cerebral artery. That will run literally through here. And then what we see here is we see the inferior frontal gyrus, pars orbitalis. We have one specific thing called the gyrus rectus, which is the most medial. You don't have to worry about that. And then, of course, we did talk about the hypothalamo-hypophyseal system. We talked about the cerebral peduncles. We talked about the middle cerebellar peduncle. And here now, in the medulla, we have these two sort of bumps. One is the pyramids, medial, and then lateral, we see these two very distinctive looking structures. And basically, remember the Latin root, both a Greco-Latin root, what grows a lot down in Greece and Rome were olive trees, and they look like olives. And of course, what we're going to learn is what we're looking at is the inferior surface of the brain. And what we're going to see is that these olives, this inferior portion of the olives, is going to be very important in terms of cerebellar communication. We'll talk about that later. And then these olives go more superiorly into the brain, and the superior olive is part of the auditory system. So we have the olives. Just get my pointer, sorry. Come on, where are we? We have the olives. We have the pyramids. We have the pyramidal decussation, middle cerebellar peduncle, cerebral peduncle, mammillary bodies, optic nerve and tract, and then the interhemispheric fissure and the inferior frontal gyrus pars orbitalis. Now, in the temporal lobe, what do we have here? We have the parahippocampal gyrus, and we have the inferior temporal gyrus. Here's the inferior temporal gyrus. Here's the parahippocampal gyrus. And then we have a bump. You notice we have a bump here and a bump here. These are things known as the temporal poles, just like we have a frontal pole and an occipital pole, at least on the superior surface. The uncus is another term for the temporal pole. So now, buried within the parahippocampal gyrus are two very major nuclei in psychology. The rostral portion of the parahippocampal gyrus has a group of nuclei called the amygdala, A-M-Y-G-D-A-L-A, the amygdala. Please capture there it is. Okay, and the amygdala is going to be very important for emotional processing into cortical sites. And we're going to talk about that at length. Then the caudal portion of the parahippocampal gyrus will contain the limbic structure, the hippocampus itself, which plays a very, very major role, of course, in initial acquisition and maintenance of memory. Now, this whole entorhinal cortex, which is the parahippocampal gyrus, plays a very important role in two very interesting types of cases. One happened in 1963. There was a gentleman who was a graduate student at the University of Texas at Austin. His name was Charles Stewart Whitman. And the night before this larger incident, he was at home and he basically shot his wife and his mother-in-law. The next day, he carried a group of rifles and things like that and went onto the University of Texas at Austin's main campus and climbed up this very large tower. And he was up at the tower and he locked the doors, and then he methodically shot 26 people to death, and about another 35 or 40 were wounded. After about an hour, police and security broke down the door and they shot Charles Stewart Whitman to death. They basically did an autopsy on Charles Stewart Whitman. What they found was a tumor the size of a small tangerine right around the amygdala. And all of a sudden, this guy was doing all types of other behaviors when they searched his apartment. He wrote in tiny, tiny print, volume after volume of material that sort of basically said he knows that the devil was coming and was going to take over the earth, and what his job in life was to save people by shooting and killing them. You know, a very, very strange type of story, but it appeared that a lot of his behavior was caused by this aberration, very large tumor in his amygdala. Then, in the hippocampus and surrounding entorhinal cortex, which is found in the parahippocampal gyrus, became another incredible case. In Canada, there was this engineer, and this engineer just could not function or work simply because he was a man in his 20s, and he was having anywhere from 15 to 30 grand mal seizures every day. And at this point, they basically found that a lot of these seizures emanated from the parahippocampal entorhinal gyri. So they did a neurosurgical procedure in which they tried to identify which side was producing the seizure. We're going to be introduced to another form of interhemispheric communication soon called the anterior commissure, and this is how the subcortical, this parahippocampal and entorhinal cortex communicate with one another on either side. So they made a guess that there was the focus was on one side, and they basically ablated or lesioned that area. The patient obviously lived, went through it, and about 3 or 4 days later, the seizures recurred. And so basically, they went in and ablated the second remaining side. This patient survived, number one. Number two, this patient no longer had seizures. Number three, he was able to sort of go back to work in some limited way. But he had one small problem. While he was doing something, or if he was doing something procedural, like an engineering task or something, he could do it. But if he was given some degree of information that he had to retain, he was incapable of doing this. A neuropsychologist by the name of Brenda Milner studied this patient over the next 60 years. He goes by the initials HM. So in 2000, I think he died in 2014. In 2012, there is a YouTube video of showing him where he's Canadian, so he doesn't have to know American politics, but he was an intelligent man, where he basically would tell HM, he would ask him, who is the president of the United States? And he'd look and he'd say, Dwight Eisenhower. He'd go, well, he was the president of the United States. But let me tell you that after Eisenhower, there was Kennedy, and then there was Johnson, and then there was Nixon, and then there was Ford, and then there was Carter, and then there was Reagan, and then there was Bush #1, then there was Clinton, then there was Bush #2, and then there was Obama, and that was the point at which he was tested. And what could HM do? You would ask him over the next 15 minutes, who are the presidents in the United States? He could repeat the list perfectly. You go away, and you come back an hour and a half later, and you ask him, who's the president of the United States? And he said, Dwight Eisenhower. What basically happened by losing that entorhinal cortex, that the hippocampus on both sides? He basically lost the ability. He had the ability to retain very short-term memory, but he had the inability to transfer short-term memory into long-term memory. And this is a very, very powerful kind of story about again, a certain degree of localization of function. So what we can see here is an enormous amount of information that we can see about the brain on the inferior surface. So now, here is the inferior surface of the brain. So again, what I want people to do is look at that, and to the degree that you have labeled all of those structures, try to label here. And if you want me to go through a review session with you, we can point out the things. So let me stop at this point and see if I have a million questions. They don't look like there's that many more questions. Oh, here, no, there's a parahippocampal. No, the parahippocampal gyrus is separated from the inferior temporal gyrus by the inferior temporal sulcus. So on this inferior surface, the parahippocampal gyrus is on the medial surface, whereas the inferior temporal gyrus is a little bit more lateral, and then it sort of goes up. Let me see if I have any other questions here. Okay, very importantly, so the whole tract is the corticospinal tract, but the other areas are just small portions of it that we identify simply where they are. Yes, that it's one continuous tract. There is never a synapse. It moves from the precentral gyrus all the way down into where it's going to go out in the spinal cord via the lateral corticospinal tract or via the anterior corticospinal tract. Very good. Let's see. So the cerebellar peduncles, in addition to having connections to those listed, yes, basically especially the middle cerebellar peduncle is a way that the left part of the cerebellum is communicating with the right side of the cerebellum, along with communicating with that large number of neurons in what is called the Grecium pontus. Okay. Okay. So now, we're finally going to leave the inferior surface of the brain and now look at the medial surface. So what is a medial surface of the brain? Literally, if we were that Freddy Krueger character and we chopped your head right along the interhemispheric fissure or longitudinal fissure and sliced completely down, that is called a mid-sagittal cut. And you can literally see the medial surface of the brain. So, what we're going to be looking at is a series of things. We're going to be literally moving superior to inferior here. So we're going to be talking about the central sulcus, the pre- and post-central gyri, a new structure called the cingulate gyrus, the septum pellucidum, the corpus callosum and fornix, the thalamus, hypothalamus, and pituitary. And then we're going to be able to see the pineal gland and superior and inferior colliculi, the calcarine sulcus, the cerebellum, and 3 means of interhemispheric communication. So, if you remember when we looked at the superior surface of the brain and the lateral surface of the brain, the central sulcus dominated. It separated the frontal lobe from the parietal lobe very, very clearly, clear as day. But on the medial surface, basically, it is going to be minimal. And let's look at that. Right now, if you were to quickly look at the medial surface of the brain, you might go, oh, there's the central sulcus. No. That is not the central sulcus. That is the superior, superior frontal sulcus. So basically, what you have here is the precentral gyrus, the postcentral gyrus, and only a minimal central sulcus. Why is that? Well, notice that if you're on the superior surface of the brain or the lateral surface of the brain, the somatosensory information that's in the postcentral gyrus cannot communicate with the motor information that's in the precentral gyrus, even though they are somatotopically aligned to one another. Why? You can't jump fibers across the sulcus. So they have to communicate somewhere. They don't communicate out at the superior or lateral surfaces. Rather, they communicate a little bit in and all the way to the medial thing. So what does this allow? The post-central, it allows, let me get my pointer here. Here we go, stop doing that. The post-central gyrus to communicate with the precentral gyrus. You can communicate there. Then we have the precentral sulcus. And now what we can see is some superior frontal gyrus right here. But we are not seeing the middle frontal gyrus or the inferior frontal gyrus until we see some inferior frontal gyrus down here. But they are seen more on the lateral surface of the brain. So now what do we have? We have another very major set of cells. It's huge. It is called the cingulate gyrus. The cingulate gyrus. Now, the cingulate gyrus is probably involved in every major sensory and cognitive and emotional process. In fact, if you're looking for a part of the brain that no matter what you're doing in an fMRI, that literally gets activated, it is usually this anterior portion of the cingulate gyrus, or ACC, or anterior cingulate cortex. This is activated during various types of cognitions. It's involved in certain types of, it's activated during decision making, it's activated during pain, and it can be activated by a wide array of sensory events. Interestingly, it is one of the original parts of the brain that made up something called Papez circuit. Papez is capital P, A-P-E-Z. And Papez was a neuropathologist working in a large mental institution. And he basically looked at the autopsy brains of long-time mental patients. And what he would basically find is selective damage in a lot of their brains that basically implicated a number of these important structures that he basically said were involved in psychopathology. And the only cortical structure that lit up here was this cingulate gyrus. The others were subcortical structures. And then about 15 years later, the initial head of the National Institutes of Mental Health, a guy by the name of Gordon MacLean, coined the term limbic system, L-I-M-B-I-C. And this limbic system basically involves whole aspects of motivation and emotion, and sort of took over a number of the structures that Papez basically described, and then expanded this further. So the cingulate gyrus is extremely important in cognition, in motivation, and in emotion. So that is, again, gray matter, a large number of cortical cells on the medial surface of the brain, just below the pre- and post-central gyri, and just below the superior frontal gyrus. Then just below that is another very interesting C-shaped structure. Notice the cingulate C is also C-shaped. This structure, however, is the corpus callosum. And the corpus callosum, rather than being cells, is large amounts of fibers, myelinated fibers. And the corpus callosum is the major form of interhemispheric communication. Again, there we go. Interhemispheric communication. So you can basically see the body of the corpus callosum, and then you can see the rostrum of the corpus callosum, and then you can see the splenium of the corpus callosum. Now, the corpus callosum, the roof of this next structure is the corpus callosum. The floor of this next structure is another fiber tract. Let me tell you that fiber tract first. This is another major fiber tract. This is called the fornix, F-O-R-N-I-X. And where is the fornix originating? The fornix is the major output of the hippocampus. The fornix is the major output of the hippocampus. And what it does is it comes out of the hippocampus and then moves superiorly, and then does again another C-shaped shape in which it goes inferiorly. This is called the columns of the fornix, and then it ends up down here in the mammillary bodies. So the fornix starts out in the hippocampus. The fornix terminates in the mammillary bodies, but there's a lot of other things going on that I'm not going to talk about now. So if we now think about the fornix being the floor and the corpus callosum being the roof, what is this structure right here? Well, immediately this structure is a thin set of cells. And that thin set of cells is called the septum pellucidum. And it's a thin sheet of cells separating the left and the right lateral ventricles. So here we're looking at a part of the ventricular system right here. And if we remove those sheaths, we would have within it cerebrospinal fluid and the whole ventricular system right in here. Okay? So, we have covered the precentral gyrus, the postcentral gyrus, the superior frontal gyrus, the cingulate gyrus. We then covered the corpus callosum, the septum pellucidum and lateral ventricles, and then we covered the fornix. Now, right here is another fiber tract. This fiber tract, instead of going rostrocaudally, is going medial-laterally, just like the corpus callosum. But it's much more restricted. You can see it's very small. The corpus callosum is very large. This fiber tract is the anterior commissure. And it's one of the three forms of interhemispheric communication. Where, if you remember in the last part of the lecture, when I was covering the entorhinal cortex, the amygdala, and a little bit of the hippocampus, the two sides of the brain, the two amygdalas, the two entorhinal cortices, and a little bit of the hippocampus communicates via the anterior commissure. Now, the anterior commissure is very, very interesting simply because it is an area of the brain that for many decades was a very popular site of something called psychosurgery. Psychosurgery originated in the late 30s and persisted all the way up into the early 80s. In certain parts of the world, like India, they still do psychosurgery, in which you're basically cutting connections between two different parts, two different hemispheres of the brain, and in order to affect some psychopathological condition. In some cases, it was successful. In some cases, it was a botch. And probably one of the more drastic types of surgery. It sounds horrible, but you can locally anesthetize the person. And basically, you take a spatula, move the eyeball out of the way, put a spatula right through the orbit of the eye, and come up and sever the anterior commissure. And for some intractable forms of schizophrenia and everything, this was a technique. I don't think it was very successful, and obviously with the advent of psychotropic drugs in the 50s, 60s, 70s, and 80s, that procedure had basically stopped. So that is the anterior commissure. So now, here is the optic tract and optic nerve and optic chiasm. Here is the mammillary bodies. So therefore, in here, what we have in this area, let me get my pointer again, is the diencephalon. The lower part of the diencephalon, of course, is the hypothalamus, and this sort of whale-looking like structure here is the thalamus. So the thalamus, the hypothalamus, diencephalon. Then what we have is the midbrain with the third cranial nerve. And now when we go into the midbrain, we're looking at this whole area right here. What we basically see is a number of very interesting structures. One, in the most posterior part of the thalamus, is the third form of interhemispheric communication. And that is called, if this is the anterior commissure, this is the posterior commissure. And it communicates between midbrain structures on two sides of the brain and parietal structures. Then, when we look at the just caudal to that, we have a very dark pigmented structure. And in the 17th century, one very famous philosopher thought that he identified the soul, S-O-U-L. That philosopher was René Descartes. When he basically saw this structure, and look where this structure is. It's sort of a midpoint between rostral and caudal in the brain, and midpoint between superior and inferior. The structure actually is the pineal gland, Pineal. And of course, it's part of the hormonal system. And the major substance that it secretes is a substance called melatonin. And the pineal gland plays very important roles in circadian rhythmicity, and it plays important roles in sleep. Now, caudal to the pineal gland, we are now in the midbrain, and we have two bumps here, right? These two bumps. And realize that we're looking at a medial surface, so the other half of the brain has another two bumps. Those four bumps known together, or protuberances, are known as the corpus quadrigemini. The corpus quadrigemini. And the two rostral bumps, one of them seen here, is also known as the superior colliculus, Colliculus. Caudal to it is the inferior colliculus. Okay. Now, notice we have the superior colliculus and the inferior colliculus. Notice that we have this channel right here. This channel below it is actually part of the ventricular system. In the midbrain, the ventricular system in the midbrain is known as the cerebral aqueduct. Okay? So literally, again, was identified back in Greco-Roman times, Romans with aqueducts, and it allowed ventricular fluid to flow from the lateral and third ventricles all the way back into the fourth ventricle, and the cerebral aqueduct was the flow of the CSF. So when people looked at Latin neuroanatomists looked at the midbrain and they saw the cerebral aqueduct, they basically said, well, anything above the cerebral aqueduct is like a ceiling, okay, ceiling, and anything below the cerebral aqueduct is like a floor, except they didn't use the words ceiling and floor. They used the Latin words for them. The Latin word for a ceiling is tectum, Tectum. The Latin word for a floor is called tegmentum, Tegmentum, tegmentum. So we have the superior colliculus and we have the inferior colliculus. They have another pair of names related to their function. The inferior colliculus is deeply involved, as you will learn. The inferior colliculus is deeply involved, as you will learn, with audition. So another name for the inferior colliculus is auditory tectum. Auditory tectum. The superior colliculus is intimately involved in vision, and I'll explain that in detail later. So that is now known as visual tectum or optic tectum. Okay? So you have visual tectum or superior colliculus. You have auditory tectum or inferior colliculus. You have the cerebral aqueduct. And now you have the midbrain tegmentum. Now, what happens is the ventricular system, as we're going to see very soon. Then the cerebral aqueduct flows into the fourth ventricle. And the fourth ventricle, the roof of the fourth ventricle is our cerebellum, and the floor of the fourth ventricle is the pons and the medulla. The pons and medulla form the floor of the fourth ventricle. The roof of the fourth ventricle is the cerebellum. So, we have covered the frontal lobe here. We have covered the cingulate. We have covered the corpus callosum. We've covered the lateral ventricles. We've covered the fornix. We've covered the anterior and posterior commissures. We've covered the thalamus and hypothalamus. We've covered the pineal. We've covered the corpus quadrigemini, superior optic tectum, inferior colliculus or auditory tectum. We have covered the cerebral aqueduct. We've covered the fourth ventricle. We've covered the midbrain, pons, and medulla and cerebellum. So we only have a couple of things left. One is now looking, here is the postcentral gyrus. If you remember, in the parietal lobe, caudal to the postcentral gyrus was the superior and inferior parietal lobules. On the medial surface of the brain in the parietal lobe, this is now called the precuneus and the cuneus. The precuneus and the cuneus. Now, if If you remember, we looked at the superior surface of the brain, and we couldn't tell the difference between the parietal lobe and the occipital lobe. When we looked at the lateral surface of the brain, we couldn't tell the difference between the parietal lobe and the occipital lobe. But finally, in the medial portion, what we basically see is a very interesting sulcus. And this is the calcarine sulcus. Okay. This is, okay. This is the calcarine. My pointer would help here. This is the calcarine sulcus. Now, the calcarine sulcus is surrounded on either side by a very important gyrus. What this is primary visual cortex. This is primary visual cortex. So, if we go back to 1978, two gentlemen won the Nobel Prize for Physiology and Medicine. Their names were David Hubel and Torsten Wiesel. What they basically identified was how visual information comes in from the eyes, goes into the ganglion cells in the retina, where you have rods and cones, and projects back through a pathway, through an area called the lateral geniculate body, and all the way back into primary visual cortex. And they neurophysiologically identified cells in the primary visual cortex that played a role in the formation, moving from a sensation to a perception, the formation of edges, where they characterized simple cells that basically measured the orientation, and complex cells that measured the length of the orientation. And basically, they started to decipher the whole visual cortex and the underlying neurophysiology, where you move from edges and dark and light, things like that, to an integrated perception through the secondary visual cortex and the tertiary visual cortex. So the big border between the parietal lobe and the occipital lobe is through these branches of the calcarine sulcus. So we see the interhemispheric communication with the corpus callosum connecting frontal, parietal, temporal, and occipital lobes, the anterior commissure that connects medial temporal lobe structures, and the posterior commissure that connects medial, parietal, and mesencephalic subcortical structures. Now, we then have somatosensory and motor systems. We're going to be talking about a dorsal column system, an anterior spinothalamic and lateral spinothalamic pathways from the medulla to the thalamus. Somatosensory is dorsal, a posterior path. Motor is an anterior path or corticospinal. And where can we figure that out is, as you probably can guess, the motor is right here is the cerebral peduncle going through the corticospinal tract. Here are the pyramids. Whereas somatosensory information is coming in a more dorsal path, coming this way up and eventually going up into the thalamus. And again, that was that thing. So now, when we have the lateral ventricles, we have circumventricular structures. What I mean by circumventricular structures are neural structures or fiber tracts that surround the ventricles. So for the lateral ventricles, the roof of the lateral ventricle is the corpus callosum and the cingulate gyrus. The floor of the lateral ventricle is the fornix. The medial portion is the septum pellucidum, and then very rostral is going to be actual nuclei called the septum itself, the lateral and medial septal nuclei. And then lateral, this is interesting. If you had a mid-sagittal cut and you peeled away the septum pellucidum and you stuck your fat finger into there and touched the lateral surface, you would be touching the caudate nucleus of the basal ganglia, a very important structure for sensory motor integration. So these are structures that encircle or encompass the ventricular system. So here we go, and by the way, this is a kind of classic slide that basically shows you something that you learned way back, I hope, in Psychology 101 or some introductory biology course, in which we basically look at the brain and we can divide the brain into two types of things based upon their coloration. One is called gray matter, and the other is called white matter. And what you can see in the brain here, with most of the structures, the neural cells and whatever, it is gray matter. And then what we can now see very clearly on the medial surface of the brain is we can see these areas of fiber tracts. So if we do the white matter, look at what we got. We got our friend the corpus callosum, a C-type structure. We got our friend the fornix, a C-type structure. We have the anterior commissure. We have the posterior commissure. We have the optic tract. And then what we can see, you can see right through here, you can see here's the corticospinal tract, very heavily myelinated. Look, here you have the middle cerebellar peduncle, another big fiber tract. Here what you can see is the corticospinal fibers running within the pons from the cerebral peduncle down to the pyramids. So you can see the pyramids, all white fiber tract. Then you can see the fiber tracts of the cerebellum. And then what you can see is the fiber tracts of the somatosensory pathways that we'll talk about later. So you can see all of these powerful fiber tracts. But then with what we can now see is the major sulci and gyri. So here is this tiny little central sulcus with the post and precentral gyrus. We have the precentral sulcus. We have the superior frontal gyrus. We have the pars orbitalis and the inferior frontal gyrus. We then have another C-shaped structure, the cingulate gyrus. Then we can either see septum pellucidum, or we can see all the way through the ventricle out here into the lateral surface, there, the caudate. And then what we see is the thalamus and the hypothalamus. Then we can see the superior colliculus or optic tectum, the inferior colliculus or auditory tectum. And then what we see is the midbrain, pontine, and medullary tegmentum. So now we're going to look at something else. We're going to look at the superior view, but what we're going to do is we're going to remove the telencephalon. And here we're going to be able to see some of those medial structures very, very clearly. So we're going to be looking at the thalamus, the medial and lateral geniculate body, and the pineal gland. We're also going to look at that corpus quadrigemini, the superior colliculus or optic tectum, the inferior colliculus or auditory tectum, and then we're going to be looking at the cerebellar peduncles, and then we're going to be looking at, by seeing the superior view, the somatosensory system as it enters into the brain. And then we're going to see yet another landmark called the obex. So, remember here, above this, here is what I'm looking at. Above this is all cortex. What we did was remove the cortex. So the very rostral pole of this is now the diencephalon. And since it's superior, there are only two structures in the diencephalon. There is the thalamus and there is the hypothalamus. By the term hypothalamus, it means it's inferior to the thalamus. So therefore, it becomes very clear that this is the thalamus. They sort of look like two hams right here, the thalamus. Now notice there's grooves in the thalamus as it goes, and it goes down laterally and inferiorly. That groove right there that you're looking at is the internal capsule. Remember, right above it would be sitting what? The pre and post central gyri. With the anterior limb of the internal capsule is the efferent fibers coming from the precentral gyrus, and the posterior limb of the internal capsule or fiber tract would be incoming fibers going into the postcentral gyrus. Now, what this allows us to do is peer down, and what we basically see right here is a very, very dark spot in here, and what we're basically looking at is the third ventricle, because what we have done is taken away, and what would be basically happening is the lateral ventricles would be pouring into the third ventricle. So we see the third ventricle, and then what we see is the thalamus, and then caudal to the third ventricle, we see the pineal gland, and then caudal to that, we see the superior and inferior colliculi. Two rostral bumps, the superior colliculus or optic tectum, and two caudal bumps, the inferior colliculus or auditory tectum. Now, here is something strange. We have the thalamus, and what we're eventually going to do is talk about 10 major nuclei of the thalamus. 7 of the nuclei are basically found up in this diencephalic area. But the other two other thalamic nuclei, the medial geniculate body, the medial geniculate body and the lateral geniculate body are called accessory thalamic nuclei, and they extend into the midbrain. Notice that the lateral geniculate body, which is more rostral here, is associated directly with the superior colliculus. Why? Because fibers from the ganglion cells are going to go and innervate the superior colliculus or optic tectum, or they're going to continue in this lateral geniculate body and eventually innervate the occipital cortex. So, if all of a sudden we had our occipital cortex removed, we would no longer get any kind of, the input from the lateral geniculate body would go nowhere. However, could we still see? And the answer is yes, because we have this superior colliculus. But what kind of world would we see? First of all, we would not see things like depth and contrast, because all we would basically see is sort of blacks, grays, and whites, and everything would be very grainy, but we would still be able to see. In fact, if we evolved from frogs, this is the end of the visual system for a frog, the superior colliculus. It doesn't have a cortex. Now, here is the inferior colliculus, and if you remember, I said that's auditory tectum because it's part of the pathway of the auditory system. And in fact, the auditory system, which is starting out in the medulla, comes up to the inferior colliculus and then goes to the medial geniculate body. And then from the medial geniculate body, that information will go to the superior temporal gyrus. So here we can see the auditory system. So now right behind that is an interesting nerve, something that I talked about at the beginning of today's lecture and the end of last week's lecture, but here it appears. This is the fourth cranial nerve, or the trochlea nerve. Remember, I said 11 of the 12 cranial nerves could be identified on the inferior surface of the brain. The 12th cranial nerve exits the brain dorsally, comes up here, and laterally. So here is the trochlea nerve. Now, whenever I look at this, I always think of it as sort of big pieces of meat. I guess that's what it is. Right here is the superior cerebellar peduncle. Right here is the superior cerebellar peduncle. Remember, the superior cerebellar peduncle is traveling between the cerebellum and the thalamus and the basal ganglia that we can't see here. And the superior cerebellar peduncle decussates. Here is the decussation of the superior cerebellar peduncle, otherwise known as the brachium conjunctivum. Here, this sort of big sort of side of ham, it looks like to me, is the middle cerebellar peduncle, or the brachium pontus. Remember, it forms a belt under the pons, but here we're looking with the cerebellum and the cortex removed. Over here we can see a fifth cranial nerve. Over here we can see the vagus nerve. And now as we go back, what we see here in the center is the floor of the fourth ventricle. And the floor of the fourth ventricle sort of comes to a chevron end right here. And that sort of end point that you see is something called the obex. It's just a landmark, just like the uncus is a landmark, or the frontal pole is a landmark, or the occipital pole is a landmark. That's called the obex. And it's sort of the end of the fourth ventricle, right here. So something else is going to happen here. I'll describe it. But then just caudal to that, in the very caudal medulla, we have another 4 bumps. Bump 1, bump 2, bump 3, bump 4. These are nuclei. The 2 medial bumps are called the nucleus or nuclei gracilis. Gracilis. This is the nucleus gracilis, and the 2 lateral bumps are the nuclei cuneatus. Cuneatus. Now, think about this. If all of a sudden I took a feather, if you remember the sort of a basketball player, Shaquille O'Neal, 7'3", if I went and I tickled Shaquille O'Neal's toe on a beach, just think how that tickling will activate neurons, sensory neurons on the toe. It has to travel up all of Shaquille O'Neal's leg. It then has to enter into the lumbar portion of the spinal cord, and it'll finally get up here in the nucleus gracilis on one side, in which you finally get a synapse of that tickling sensation, a somatosensory sensation. So the nucleus gracilis and the nucleus cuneatus, we're going to see how they are the integral first synapses of a very fast somatosensory system. So here, let's again look at the thing, and again, what you get is a beautiful appreciation for gray matter and for white matter. So what you basically see here is the thalamus, and you can see a bunch of white matter, but also on that outer band, what we can see here is the posterior and anterior limbs of the internal capsule. Then we can see the gray matter. Then you see absolute black. That's the third ventricle. You see the pigmented pineal gland. Then you see the superior colliculus or optic tectum. You see the inferior colliculus or auditory tectum. You can see the trochlear nerve coming out the dorsal surface. Then you can see again white matter. Remember our friend, the superior cerebellar peduncle, or brachium conjunctivum, is sets of fibers that are ascending and descending between the cerebellum, the thalamus, and the basal ganglia, and that there's a decussation here. Then we see the middle cerebellar peduncle, or brachium pontus, a huge white matter tract that is enveloping the inferior surface of the pons. And in this actual picture, you can see the sort of little depression in here, because what you have in here would be the fourth ventricle. And here we can see the obex. And then what we can see, and not clearly, but we would have the gracial nuclei medially and the cuneate nuclei laterally, part of the somatosensory system. There is the inferior view without cortex. I don't have to go through this because it's exactly the same thing as what we saw. Here we would see only 10 cranial nerves. We don't see the trochlea because that exited dorsally. We don't see the olfactory because we removed the telencephalon. Everything that's here was covered on the inferior surface. Then we finally have the lateral surface without the cortex and the cerebellum. And I sort of love this picture because what it sort of reminds me of, I was a great aficionado of monster movies of the 1950s. I was born in 1946, and with a group of my friends and everything like that, I think when we started, when we were 7 or 8 years old, our parents would send us out as a group. We'd all go up on a Saturday afternoon and watch movies. And of course, what this picture always reminds me of is an interesting classic called A Creature of the Black Lagoon. But in any case, we have removed the cortex and the cerebellum, but now we're looking from a lateral view, not a superior view, not an inferior view.
Professor?
And looking at a lateral view, we're seeing, here is the superior surface of the brain, here is the inferior surface of the brain. And what you get a sense of right here is how a lot of the fibers and things that are going in and out of the brain sort of go directly. So, of course, if we don't have a telencephalon, here is the thalamus, and then here is the internal capsule. Then what we have is the optic nerve, the optic chiasm, the optic tract, the lateral geniculate body, and if we ever followed, it would go right back to the occipital cortex. So there's the optic nerve. Here is the infundibulum and the pituitary stalk. Here.
Professor Bodnar.
Yes.
Me. Hi.
Yeah.
We have 4 more minutes. Would you mind?
Yes, I know. I'm going to cover this again. I just want to finish the thought. Go ahead.
Can you check the chat before we have to end?
Sure, we can. And by the way, what I'll do is I will check every one of those chats, put them down, and cover them at the beginning of the next class.
You
okay?
can't actually save the chats. So
what's that?
The chats disappear as soon as you end the Zoom.
Oh, I know that, and I won't let them disappear. Don't worry, I'll take them down. I'll look at every chat since 11 o'clock. Okay? I will not allow them to disappear. Okay? So, here we go. I see well, let me just, okay, lateral geniculate, I'll show you that the next time. The parieto-occipital sulcus and the calcarine sulcus are 2 different sulci. I wanted to cover more the calcarine sulcus, but you can see on the medial, the whole point here is you can clearly differentiate the occipital lobe from the parietal lobe only on the medial surface of the brain, and the 2 major sulci that do that are the parieto-occipital and the calcarine sulci. So yes, that's an important thing, and you can save them. Okay, the file button does that. But I think I answered every other one of the chat questions, but we will certainly go through that again when we go here. So what you'll be getting from me is another email, and what I'll try to do, and I'm going to ask all of you, I see the 3 dots, all of you to maybe begin to chat with one another as to when would be some very good times for asynchronous reviews, okay? The unfortunate thing is when I go into file sharing, all I'm seeing is 2 or 3 people on the side, and I'm not seeing too much, so I'm not seeing too many reactions that I would react to in in-person meetings, but we can go and do all of these things, okay? So, oh good, you're going to make a group chat for this class if people are down, very good, and you're on WhatsApp, so you'll set up study groups, and you can set up hopefully common times, so I'm not doing 30 extra hours where we can have some review sessions, okay? So we will work on this. Now, let me again say this. So if you basically looked at my schedule, you're going to go, oh, Bodnar, you're 2 lectures behind. We haven't covered the ventricular system, and we haven't covered the blood supply. I am covering things at a given rate. For some of you, you may be still considering this is far too quick. It's an enormous amount of information, but I can't give it any other way. I mean, boy, the famous quote of Donald Trump, It is what it is. I hate to sound like that, but when I'm showing you the superior surface of the brain, I can't say, ignore this, pay no attention to this. I have to name a number of things, and we work through, and especially when we got to the inferior surface of the brain and the medial surface of the brain, you're going to have a series of questions, and then the superior view without the cortex, you can have another set of questions. But just realize I'm showing you this for the first time to give you a name, and then the whole rest of the course, once we get beyond the spinal cord, we're going to be going through the brain systematically, step by step, slice by slice, quarterly to rostrally, so that you're going to get the same information over and over again and elaborate it. And the point of the matter is, I found that if you gave this course and all I ever did was just name the neuroanatomical structures, you would be brain dead sleeping on the desk. What I try to do, because virtually all of us at the end of the day are in psychology, which is the study of behavior, and here we're trying to do brain behavior relationships. I'm trying to put those things all together. So what I see right now is I'm 2 minutes over, 2 minutes after 12. I'm a horrible liar. I didn't stop. This is sort of, I am what I am. I'm Popeye. I am what I am, and whatever. So we will work on this. Please communicate with me when you think you have some critical things of information, and I can actually start setting up these asynchronous meetings further down the line. I do want to thank, I think her name, Anda Esteron, for interrupting me at this point and breaking in. You are a very brave person. Thank God. Back in the old days, when I talked a lot faster and a lot longer, one time I was teaching as a senior graduate student, I was teaching a experimental course, and I kept on talking, and it was a class of about 15 people. They were in the room with me at that very moment, and somebody raised their hand. They asked the question, and I answered the question, and then they said, "Oh, by the way, do you know it's 1:25?" I go, "No." The class ended at noon. Nobody opened their mouth to say, "Stop already," or got up and walked out. So thank you, Anda, for stopping me, although I was always able to see I was in the last couple of minutes. Okay? I'm going to be repeating and repeating and repeating and repeating. And again, in these reviews, if you have a question, ask it and talk among yourselves and end up learning, knowing what you don't know, and then being able to ask the questions and go on. Because I'm hoping, again, this is the very basis of the language that you're going to be using throughout your careers if you're in this area of neuroscience and behavioral neuroscience. So, have a very good day, decompress, rest, and we'll move on. Okay? So, have a good week.
Thank you.
Thank you.